Okay, Quiz Monday will be over enzymes, which we will get through most of that today, if not all of it. So please be prepared for that. And then also read part 1 of your lab on enzymes, and we'll start the first part of a 3-part lab on enzymes. So everything's kind of coming together right now. Any questions? Okay, here's an energy profile for a hypothetical reaction where we've got 2 molecules that are going to be converted into 2 different products. Okay? So what does this particular energy profile tell you about the reaction? Is it exergonic or endergonic? It's exergonic. How do we know that? Exergonic reactions are spontaneous. They tend to go from states of higher free energy to lower free energy. The delta G for this particular reaction is less than 0. Why? Because we take the free energy of the products minus the free energy of the reactants. That will give you a negative value. These molecules are unstable. These are more stable. And so this law is behaving in accordance with the second law of thermodynamics, which says that things spontaneously achieve a state of greater stability. That make sense? Okay? So, good time. What has to happen to get from here to here in terms of the bonds that are joining together these substances? What's happening to the bonds at A and B, C and D? There's some bond breaking involved, isn't there? Okay, doesn't the bond between A and B have to break? And the bond CD have to break in order for reaction to take place? What else happens? Bonds are broken, they're also formed, right? So new bonds are formed now between A and C, B and D. So chemical reactions involve bond breaking and bond formation. Which one requires energy, breaking or forming? Bond breaking requires energy. Okay? Bond formation releases energy. Well, in reality, if you look at the energy profile of this particular reaction, it doesn't look exactly like I've drawn up here. Instead, it looks like this. Okay, so here's our reactants. Here's our products. Higher free energy, lower free energy. So that free energy difference between reactants and products represents the delta G. It's a negative value because we take this minus this. But what's this in here? What's this little addition? What does that hump represent?
Activation.
It represents something called the activation energy barrier. Here, the bonds are intact. But for a reaction to take place, these bonds have to be broken. Bond breaking requires an input of energy, right? So at some point in time, these molecules have to absorb enough heat energy from their surroundings, which causes them to collide more frequently and bump into one another more, which will put more stress on those bonds. And at some point in time, those bonds become sufficiently stressed that they're ready to break. And then once the bonds break, then we proceed downhill, and as new bonds are formed, energy is released. Okay? So this represents kind of the energy hurdle that must be gone over to get from here to here. Does that make sense? If this were DNA, DNA is a very highly ordered molecule, very high free energy. Can DNA break down into its nucleotides? Can it be hydrolyzed into its A, T's, G's, and C's? Yeah, I mean, that's part of the digestive process, for example. Okay? Can ATP be hydrolyzed into ADP and phosphates? Yeah, we hope so. Okay. Which one of those two, DNA or ATP, do you think will have a higher energy of activation barrier? Which one will fall apart more readily at the same temperature? ATP. Why? Because we want that conversion, or the cell wants that conversion to take place from ATP to ADP and phosphate. Does the cell want its DNA to fall apart? It will create some problems in your chromosomes, right? So the barrier height will differ depending upon what molecules are over here as reactants. DNA molecules, protein molecules have a pretty high energy of activation barrier. ATP molecules have a pretty low energy of activation barrier. Why does ATP have a low energy of activation barrier? Why is this low? Because remember that terminal phosphate group is very unstable, very fragile, and is easily broken. So you don't need to have that much thermal energy in your surroundings to make those molecules collide enough to break the bonds. Is it important sometimes for organisms to take a molecule like DNA or protein that has a pretty high energy of activation barrier, but still want to break it down? Are there instances where you need to break down proteins? Or DNA? Or carbohydrates? Or lipids? Yeah, when you're trying to use them as food sources. But if this energy of activation barrier is high, is the cell ever going to have enough thermal energy to really make that happen? No. Now, theoretically, what could you do to make that reaction happen faster? You could catalyze it, or in the absence of a catalyst, what's another way you can make chemical reactions occur faster? You can heat them, okay? The more you heat them, the more energy can be absorbed from the environment, the more collisions that take place, the more bonds that are stressed, and therefore, the more internal energy you have to get over this particular barrier. Can cells use that strategy to increase the rate of necessary chemical reactions by just heating themselves up? What would be the problem with doing that? Okay, that's one possibility, is it would speed up all the chemical reactions, not just that one.
Think about the cell's own proteins to denature.
Yeah, then you could start, if you get too hot, then molecules start denaturing. DNA starts denaturing, proteins start denaturing. So increasing rate of chemical reactions in the cell by increasing temperature just really doesn't work. And remember that cells and organisms kind of try to maintain a stable temperature. They don't want a big fluctuation back and forth. Okay? So, this reaction, this profile right here tells you about whether the reaction is spontaneous or not. Whether it is thermodynamically favorable. Does this particular energy profile say anything about the rate at this reaction occur? Just because a reaction is spontaneous, does it mean that it goes fast? No. Spontaneous or not spontaneous is kind of like stop and go. But rate has to do with speed. And those are two different parameters that we need to consider. So just because a reaction is potentially spontaneous, if this barrier of activation is really high, then the rate of it is going to be really, really slow probably, unless we heat it up or unless we catalyze the reaction, unless we add a catalyst. So what we want to talk about, particularly in context of what you're going to do in lab for the next few weeks, is talk about the role that enzymes play in catalysis of metabolic reactions. So what do you know about enzymes already? What can you tell me that you already know about enzymes?
It's a catalyst.
Okay, they are catalysts. Can you be a little bit more specific in terms of what kind of catalysts they are? For example, platinum is a catalyst. Is platinum an enzyme? Platinum is an inorganic molecule. What are enzymes? They're proteins. They're organic molecules. So they are organic catalysts. Okay? So all enzymes are proteins then, correct? Very few absolutes in biology. There are some exceptions to that rule that all enzymes are proteins. Most of them are. The vast majority are. You know, Brittany, which ones aren't?
No.
Okay. Anybody know? Rick?
Are there some RNA molecules that are?
Yep. There are some RNA molecules that are catalytic called ribozymes. And we will have a chance to look at ribozymes a little bit later on when we talk about protein synthesis or translation. But we're not quite there yet. So most enzymes are proteins, but there are a few exceptions to that. What else you know about enzymes? You got proteins and catalysts.
They have an active site.
They have this region called the active site. Now, think about this for a second. An enzyme is a single or sometimes multiple polypeptide chains. Remember what a polypeptide chain is? It's a bunch of amino acids strung together. Okay. And then what do polypeptide chains do? Do they just stay linear like this? They fold up into complex three-dimensional structures. They have secondary structure. They have tertiary structure. Many enzymes have quaternary structure as well. They're made up of more than one polypeptide. Well, when they fold up, this little region is created called the active site. And what happens at the active site?
Is that where substrate binds?
Okay, that's where substrate binds. And that's really where the catalysis occurs. It's like a little pocket within that folded up structure. What does all the rest of that polypeptide chain do that's not part of the pocket? It helps to kind of form the structure that then defines where the pocket is. So as the polypeptide chain folds up, that kind of defines what the pocket looks like. Okay, so active sites are important. We'll talk about those. What else do you know about enzymes? What's a characteristic of catalysts? We said that they speed up reactions. What's another characteristic?
They start reactions.
Pardon me?
They start reactions.
They used to, they facilitate and speed up the rate of a reaction. But what happens to that catalyst during the reaction?
It's maintained.
It's maintained, it's not changed, it's regenerated. So the catalyst isn't used up in the reaction. It's regenerated. It can be used over and over and over again. Now, can enzymes last forever? No, they're proteins. And all molecules in the cell eventually will degrade or turn over, if you want to use that term. But some enzymes hang around for quite a long period of time. There are, however, factors that can cause enzymes to denature. And what happens to the active site if the enzyme denatures? change that active site? Yep. Change the shape of the enzyme, denaturation, then you change the shape of that active site. Let's go back to the active site for a second. Can just anything bind to that active site? There is a specificity between the shape of the active site and the shape of the substrate. Much like there is a specificity between receptor molecules on the surface of a membrane in cell signaling and signaling molecules. Or much like there is a relationship between membrane transport proteins and the molecules to which they bind and transport across the membrane. So specificity is very important in enzymes. Certain enzymes bind to certain substrates. Know anything else about enzymes? Okay, this shows you what is typically referred to as the catalytic cycle of an enzyme. So here we've got an enzyme molecule. This is a polypeptide chain folded up into a particular three-dimensional shape. And this represents the active site of that enzyme. That's where the substrate, or sometimes more than one substrate, will bind. Okay? Some reactions have a single substrate. Many reactions have multiple substrates. The one that you're going to do in lab next week has multiple substrates. It's a two-substrate reaction. But the shape of this has got to be proper to fit the shape of those substrates. There's a complementarity between binding to this and the shape of this. What's the analogy that's sometimes used between the interaction of an enzyme and a substrate? Have you ever heard of the lock and key concept? The substrates in this particular case would be keys. The enzyme would be a lock. And when you stick the proper key in the lock and turn it, the door opens. Or catalysis occurs in that particular case. That's an okay concept to use for right now. But as it turns out, that lock and key model is kind of inadequate to really explain how enzymes work. Because what typically happens to enzymes when something binds to them? They change shape, right? Remember when you phosphorylate a protein, it changes its shape. When you dephosphorylate it, it changes its shape. So is it likely that when these substrates bind to the enzyme, that the shape of the enzyme will change? Yep. And that actually, as it turns out, can be an important part of how this catalysis occurs. Okay, eventually a chemical reaction occurs as a result of bond breaking and bond formation that results in the substrates being converted into products. Products have a different shape than the substrates did. Therefore, they won't tend to bind to those sites any longer in the active site, and the products will be released. What can the enzyme do again? Pick up some more substrates and do it again. Again. It's not atypical for an enzyme to be able to do 1,000 of these catalytic cycles in a second. Grab a substrate, make product. Grab a substrate, make product. That's pretty fast. They dramatically increase the rate of the chemical reaction. These substrates without the enzyme might sit there for years and really never interact because their temperature is fairly low. Maybe they're at room temperature, maybe they're inside a cell, but the energy of activation barrier is very high. So they just won't interact with one another. Thermodynamics says they can interact. They've got a negative delta G. But the rate of the reaction is so low because of the low temperature. But in the presence of the enzyme at that same temperature, they just go like that. Okay? So you would see that in lab next week. We're going to do an enzyme called peroxidase. And we're going to mix together a couple of substrates, hydrogen peroxide and guaiacol. And if you pour those into test tubes at room temperature, they're going to sit there forever and never form a product. The way you can tell a product is formed is the two original solutions are colorless, but the product they form is brown. And we can measure the intensity of the brown color with the spectrophotometer. But at room temperature, they'll just sit there. That tube will never turn brown. But add some extract from turnips, which contains the peroxidase enzyme, and bang, now it starts really going fast. And the amount of brown color that's produced will be very dramatic in a short period of time. This shows you an enzyme in a little bit more kind of detail in this kind of molecular ball and stick model, giving you an idea of kind of the complicated three-dimensional shape of the enzyme. And here's our active site here, a little pocket where the substrate can bind. What actually happens when the enzyme and substrate bind is that the enzyme shape changes and really becomes better fit and kind of surrounds that particular substrate and really tugs it in there very tightly and really makes a very, very complementary fit to that substrate. That particular phenomenon is called induced fit. So as a substrate bind, it kind of makes the enzyme fit the substrate even better. Okay, so enzymes are catalysts. How can we explain the role that enzymes play in catalysis? One of the things that's very, very important to understand is enzymes do not alter the thermodynamics of the reaction. They cannot magically make a non-spontaneous reaction spontaneous. They cannot change the delta G of that reaction. Because delta G is just a reflection of the difference between free energy of the reactants and free energy of the products. Enzymes can do nothing to change the free energy of either one of those molecules. So how can they increase the rate if they don't change free energy? What enzymes do is they lower this energy of activation barrier. When you combine the enzyme with the substrate, things happen so that this barrier is now much lower. And it's much easier to get over that barrier than it was before. So even at normal cellular temperature, without an enzyme, this barrier is so high that these molecules just aren't colliding enough and strongly enough to get over that big barrier. But at the same cellular temperature and the same rate of collision, if this barrier is much lower, then more of those molecules are going to have sufficient energy to get over that little lower barrier. Any of you ever run the hurdles before? Any track people in here? Okay. Over the same distance, is your speed going to be higher in low hurdles or high hurdles? You can probably run the lows faster, right? There's a lower barrier you've got to get over. That's kind of the same thing that's happening here in these chemical reactions. In the presence of an enzyme, this barrier is lower, and therefore it's easier for more molecules to make it over that transition hump and then start moving down to forming products. So the rate of the reaction will occur much faster in the presence of an enzyme than in the absence. Now, the book talks about 3 or 4 different ways in which enzymes can lower this barrier. I just want to mention one of them right now to kind of give you some context. Remember the transition state, the bonds are very fragile and ready to be broken. And then when they break, the reaction occurs. Remember that substrates attach to enzymes at the active site. And that causes a change in the shape of the active site. And remember, the substrates are bound there. So if we've got these substrates bound to this active site, and all of a sudden the active site starts changing shape, is that not going to kind of twist those molecules and distort them? And as it twists and distorts them, will that not make those bonds more fragile and more easy to break? That is one of the ways in which enzymes catalyze reactions, by stressing bonds when the enzyme binds to the substrate, making the bonds more fragile and effectively lowering that transition state. There are other ways too, but that's not particularly important at this level of our course. Okay? So, here's a comparison of a catalyzed reaction and a non-catalyzed reaction. Here's the non-catalyzed reaction here in black. Here's the catalyzed reaction here. Note that the delta G is not changed. The only thing that's changed is this energy of activation barrier is lowered. So now more molecules have enough internal energy from the environment to get over that hump and react. Well, one of the things we're going to do in lab is to look at some environmental parameters that affect enzyme rate. And let me throw out a couple of terms here that you want to be familiar with. When you hear the term enzyme activity, what we're talking about is rate. How fast will the enzyme catalyze that reaction? How quickly will substrates be converted into products? How fast does this occur? A rate is just an amount per time. Miles per hour is a rate. Okay? The amount is miles, the time is hours. We're going to measure enzyme activity by measuring how much product is produced, brown colored stuff, and how are we going to quantitate that with spectrophotometer. The more brown stuff, the higher the needle will rise on the spec. And we can measure time with a watch. So we're going to measure the amount of product produced in a given amount of time, and that will give us a rate. And as I said, the rate of the reaction will be much, much faster in the presence of the enzyme than in the absence. But we're going to go a step further. What kind of other factors influence how fast this reaction will go? Some groups will work on the effect that substrate concentration has on the rate of the reaction. Some groups will look at the effect that enzyme concentration has on the rate of the reaction. What other kind of factors do you think could influence enzyme activity besides substrate and enzyme?
pH.
pH? Another group will do pH. What's another factor that might influence it? Temperature. And then, one lucky group will get to do some work with enzyme inhibitors. The way we're going to do things is you're going to learn next week how to do the basic enzyme assay. And we'll start you out first with learning how to pipette everything and getting the reaction set up, using the spectrophotometer to measure the rate of the reaction. We'll have you do manual timing, and so you will look at a watch and take readings from the spec every 30 seconds. And we're going to see if we can get any kind of replication of results doing the same experiment over and over again. We'll do that a couple times. Then we'll show you how to use a software device, LoggerPro, which you've used. And basically, that will collect your data for you and graph it in real time. And then once you've got that graph, you can do some interesting analysis of that. At the end of the period, we will have a little contest for optional points where we'll see which group can get the most reproducible results for at least 2 trials of the same experiment. And ideally, your rate of reaction would just be identical in both cases. It's not going to happen that way every time. But we usually end up having pretty good competition. Students kind of get into that competitive aspect of it. First week, once you learn how to do the basic enzyme assay, then you will be assigned your parameter the next week, and you and your partner will investigate one of these aspects. Okay? And there will be some additional instructions in your lab test. Then the following week, you're going to get together and kind of pull your stuff together so you can do a presentation, which is after we come back from break, right? And you'll present your results to the other groups because you'll learn some interesting things about this enzyme from the other groups. And their results, you'll kind of go, oh, wow, that really helps to explain my results. So this is going to be a fairly extensive exercise. What do inhibitors do to the rate of an enzymatic reaction?
Slow it down.
They slow it down, right? Inhibitors will bind to the enzyme. Some inhibitors bind to the enzyme covalently. That's a very strong bond. And once it's formed, it's hard to break. If you have an inhibitor that binds to an enzyme covalently, it's pretty much irreversible. That inhibitor binds and it's stuck there. And so the enzyme is pretty much permanently knocked out then. There are a number of metallic compounds that bind to enzymes irreversibly and basically denature. Things like lead and cadmium and mercury. Those substances are all potential poisons. The reason that they're poisons is that they bind to your enzymes and knock them out. There are also some other poisons that you might have heard about. Anybody ever heard of sarin? Sarin's a gas that was used in 1995, I think, in Tokyo. It's kind of a nerve gas. It binds competitively to an enzyme that's involved in the nervous system. You've all heard of DDT before, right? DDT is a irreversible competitive inhibitor of some key enzymes in insect nervous systems. And by inhibiting those enzymes, it kills the insects. Some inhibitors bind to enzymes by non-covalent bonds, like hydrogen bonds, for example. And those are much weaker bonds. So the binding of these inhibitors can be reversible. If there's a lot of inhibitor presence, then there's going to be a tendency for the enzyme to bind. If there's not so much inhibitor presence, then not as great a tendency. So inhibitor concentration really plays a particular important role in this reversible binding. Well, there's two important types of reversible enzymatic inhibition. They're called competitive inhibition and non-competitive inhibition. In competitive, the inhibitor binds to the active site. What else is trying to bind to that active site? Substrate is. So there can be a competition between the substrate and the inhibitor, both trying to get the same place. If there's a lot of inhibitor and a little substrate, then most of the enzymes will be bound by inhibitor, and they won't work. If there's a lot of substrate and little inhibitor, then most of the enzyme will pick up substrate, just by chance. And the reaction will proceed somewhat. In non-competitive, the inhibitor binds to a site other than the active site. Remember what prefix means other? Prefix allo can mean other. So these sites are oftentimes referred to as allosteric sites. What happens when that inhibitor binds to the allosteric site? It changes the shape of the enzyme. And if the shape of the enzyme changes, could that change the shape of the active site? Yep. And would that affect catalysis? Yep. So here's our normal binding. Here's our enzyme. Here's our active site. Here's another site down here, an allosteric site. The substrate fits the active site, it binds, and catalysis occurs. In competitive inhibition, there is some inhibitor that looks a lot like the substrate. Structurally, they're pretty similar. And so this can bind to the active site as well. But if this binds, does the enzyme catalyze a reaction? No. So the ratio of substrate to inhibitor will really determine the rate at which the enzyme catalyzes this reaction. Whole lot of substrate, little bit of inhibitor, pretty much unchanged reaction. Start getting more inhibitor relative to substrate, and more enzymes start getting knocked out. And eventually, if there's enough inhibitor relative to substrate, pretty much all the enzymes are occupied by the inhibitors and nothing really happens. Here's non-competitive inhibition. Okay? Here's our substrate. Here's our non-competitive inhibitor binding to this allosteric site. When that binds, it changes the shape of the enzyme, which alters the shape of the active site, and consequently, substrate won't bind. As long as there is inhibitor around, does it matter how much substrate is present? Will increasing substrate concentration affect or overcome this inhibition as long as this inhibitor is around? No. Doesn't matter how much substrate there is if this is a wrong shape. So in competitive inhibition, you can kind of overcome inhibition by swamping out the inhibitor with substrate. So you add a whole bunch of substrate and eventually you can reverse the reaction. But in non-competitive inhibition, as long as there's inhibitor present, it really doesn't matter how much substrate is there. Okay. There's a very practical application to inhibition, particularly this non-competitive inhibition. This particular type of inhibition is called feedback. Feedback inhibition is a mechanism that the cell uses to regulate metabolism and to help achieve homeostasis. You've all experienced feedback inhibition before. It's a very kind of simple process whereby you've got a process of some type that makes a product. And the product, as it builds up, shuts off the process that makes it. It's a self-correcting phenomenon. Where have you experienced this before? In any place you've lived where there's a heating system that works properly. Ideally, what would you like to have happen with your heating system? You would like to have a relatively constant temperature level in that room, defined by what you set the thermostat to be. What produces the heat? A furnace. Okay? So if the temperature drops below the thermostat setting, that sends a signal to that furnace, put out heat. But at some point in time, the heat that's produced causes the thermostat to rise, and that sends a signal back to the furnace, shut off. Okay? So you have this slight fluctuation of temperature up and down as your furnace turns on and turns off. But what really governs it is the effect that the heat has on the thermostat. That's what controls that homeostatic system. Well, when we talk about inhibition in the context of poisons, that probably leads you to believe that anytime you inhibit an enzyme, that's bad. But there are instances where it's important for the cell to temporarily shut off an enzyme. Particularly if it can do it in such a way that it's reversible, so it can turn the enzyme back on again. Think of this analogy. When I showed you that metabolic map of all the cell's chemical reactions, I said it looked like kind of a crazy street map. Well, think of metabolism in that particular context for a moment. Okay? In a city, there's all kinds of different destinations where people want to go. In the cell, there's all kinds of different kind of chemical reactions that have to occur to make that cell work properly. And in some situations, it's making one thing. At other times, it's making another thing. In cities, there might be flow of traffic that differs from one time of the day to another. Maybe there's an event, and you want traffic to flow one way. Maybe when people are getting off work, you want to facilitate traffic flowing another direction. Okay? One of the ways that you can manage traffic flow in a city is through stoplights. Okay? If you want to get someplace fast, and you know there's a route where you're going to run into pretty much red lights all the way along the way, you're not going to take that route, are you? You're going to follow the route where there are the most green lights. Well, that's kind of what enzymes do to metabolism. When they're active, they speed up reactions, green lights. When they're inhibited temporarily, they slow down reactions, red lights. So the cell can start to kind of direct its metabolism by turning on enzymes at some point in time and turning other enzymes off at other times. And that helps to make sense of the cell's metabolism or traffic flow. Well, one of the ways it can do that is through this phenomena of feedback inhibition. So, last thing I'll talk about, and then we'll finish up on Tuesday. This represents a very simplistic set of metabolic pathways. And so we've got some compound here, L. And one metabolic pathway could be L to M to O to P to Q. That's one set of reactions. So here's some product. Here's another potential pathway, L to S. Here's another pathway, L to M. At some point in time, the cell might need a lot of M. At other times, it might need a lot of S. At other times, it might need a lot of Q. Or maybe Q and N at the same time. Or maybe all three of these. Okay? There's a lot of different possibilities. In feedback inhibition, what can happen is the product of one reaction can go back and temporarily inhibit an enzyme that's an early step in that reaction. So if you've got a lot of Q already, do you want to waste your L resources making more Q? No. But what can happen? As Q builds up, that inhibits this enzyme. So what's L going to tend to do now? If these are working, that's going to be formed, and that's going to be formed. What if you've got a lot of S already? Want to use your resources making more S? No. But as S builds up, that can competitively inhibit this enzyme right here. So there's a block here, a red light. So now what does L do? It might go this way, or it might go this way. So as end products build up, if they can temporarily slow down an enzyme involved in the production of that product, that gives you a way to regulate metabolism. So these feedback regulation mechanisms are really, really important to the cell. Okay, we made it through most of the stuff that's in the section for the quiz right now. So if you haven't read the quiz, make sure you read that particular section. We will also give you back lab handouts from the cell membrane lab. And don't put those away when you get them, because we'll be going over that lab, and particularly the yeast transport experiment, because some of you didn't seem to know what was going on there. So we're going to show you some pictures and make sure you can go through the logic of that, because that will be on your lab quiz. So I'll also give you a study guide for the lab quiz on Monday. Okay? So have a good weekend. And don't forget exam corrections, okay? If you're going to go for those extra points.
Are we allowed to have the page of notes?
Yeah, page of notes for the quiz on Monday. Yep, you can always do that.